Hello friends, today we are going to start about Krebs cycle. So before going to the Krebs cycle, first I want to make you understand that normally it is an aerobic process. For example, I have taken this as the cell. So within the cell, we do find the cell organelle where the Krebs cycle takes place is called as the mitochondria, and this part is the mi- mitochondria. Got it? So normally, glycolysis is a common pathway, glycolysis common pathway. So already the glycolysis has been taken place in the cytoplasm, and the end product is pyruvic acid. So this pyruvic acid is unable to enter into the mitochondrial double membrane, that is the inner membrane. So this part is the inner membrane where the mitochondria, where the pyruvic acid is unable to pass through. It's unable to permeable. So what happens now here? What happens and get a link reaction takes place, and this links reaction due to this pyruvate dehydrogenase complex. This pyruvate dehydrogenase complex helps this reaction, and this pyruvate dehydrogenase complex contains the enzyme that is dehydrogenase, that is decarboxylase, and also that is transacetylase, and also acetylation complexes of Mg2 plus thiamine pyrophosphate and lipoic acid. So these are the total enzymes, and the enzyme complexes present together, we do call them as pyruvate dehydrogenase, which is a complex reaction, and just it takes place in the normally this reaction takes place inside the inner membrane, and next that is to the matrix. In the matrix part of the cell membrane of the mitochondrial membrane, it is found to be taking place. So before entering into first, we know that this particular part is regarded as the link reaction, that links with, that is glycolysis, to, it links with to what, now that is Krebs cycle. So in this link reaction, before going to this link reaction, first we know details how the reaction takes place. So normally this is the pyruvate, which is a 3-carbon compound. It is a 3-carbon compound, carbon 1, carbon 2, and carbon 3. So with the help of the coenzyme, and this is the coenzyme having sulfate within it, thiosulfate, and in presence of this coenzyme removes, when it enters into the pyruvic acid, it removes carbon dioxide. So normally the entry of the molecule of pyruvic acid as the end product of glycolysis is 2 molecules. So what happens here? 2 carbon dioxide is being released out, but simultaneously the NADH2 is also being released out by the entry of NAD. So here dehydrogenase takes place as well as decarboxylase takes place. So with the removal of 1 carbon dioxide, the compound form that is called as acetyl coenzyme A. And this acetyl coenzyme A form is a 2-carbon compound. It is normally a 2-carbon compound. So it enters, it is in the matrix part. So this all taking place is in the matrix part. So what happens as it enters into matrix, the oxaloacetic acid, which is a 4-carbon compound, this reacts with this acetyl coenzyme A. So both of them react. This is 4-carbon, this is 2-carbon, together it forms 6-carbon compound, and this 6-carbon compound present in the enzyme that is called as citrate synthase. And by entering into this coenzyme is being given out, is being removed out, and it again moves up into the pyruvic acid to continue the cyclic reaction. So this is a cyclic reaction, this part. So now what happened during this reaction? The water is being used up. So with this entry of water and the acetyl coenzyme A and oxaloacetate, all this takes place in the matrix of the mitochondria, and within this matrix 6-carbon compound form that is called as citrate or citric acid. So it is called as citric acid cycle. And this citric acid cycle contains the 6-carbon compound, and it contains 3-carboxylic acid. So it is called as TCA cycle, that is tricarboxylic acid cycles. So what happens here? What happens in presence of an isomerase reaction takes place in presence of the enzyme that is called as aconitase, and this aconitase removes the water molecules. With the removal of water molecules, this citrate converts into cis-aconitic acid. Only isomerism reaction is going on. Again, this water molecule that is released out is again taken in presence of the same enzyme that is called as aconitase to produce the compound that is called as isocitrate, and both of this compound are of 6-carbon compound, as we know. So this isocitrate undergoes dehydrogenase reaction in presence of the enzyme isocitrate dehydrogenase, and what happens, an NADH2 is being produced, and NADH2 is given out with it also a 6 carbon compound, of oxalosuccinic acid is produced. And this oxalosuccinic acid undergoes decarboxylation. So this is called as decarboxylation with the removal of carbon dioxide. So with the removal of carbon dioxide, this oxalosuccinic acid converts to a 5 carbon compound. It converts to a 5 carbon compound that is called as alpha ketoglutaric acid. That is called as alpha ketoglutaric acid, which is a 5 carbon compound. So now this 5 carbon compound, what happens? It again undergoes a link reaction, because this part is called as the link reaction, that is within the matrix and within the cycle. Here one link reaction, so the same complex is used, that is called as dehydrogenase complex, alpha keto dehydrogenase complex. And what of the complex used in the pyruvate dehydrogenase? The same enzymes, the same enzyme complexes, all are used within this. And with this, what happens, the coenzyme is also used. And when the coenzyme is used and carbon dioxide is given out, when carbon dioxide is given out, and it converts to a 4 carbon compound, that is called as succinyl coenzyme A, which is a 4 carbon compound. And here the link reaction takes place, that is 5 carbon compound then converts to 4 carbon compound in presence of this alpha ketoglutarate dehydrogenase complex. This alpha ketoglutarate dehydrogenase complex also contains the same enzymes groups that is used in the link reaction. So this is a complex reaction having the same reaction. So both of them, this part and this part, both of them have the same enzyme complex reaction. Here 3 carbon compounds converts to 2 carbon, here 5 carbon converts to 4 carbon. And here what happens in this reaction, in the presence of thiokinase succinyl coenzyme, a cyclic reaction takes place that is been found in the pyruvate link reaction. So this 4 carbon compound succinate dehydrogenase, a succinic acid, now having a coenzyme within it. The coenzyme is called as succinic coenzyme A, which is 4 carbon compound. So in presence of GDP and P, that is it given now GTP, that is the substrate level ATP. Here the first substrate level ATP is produced, that is called as substrate level ATP. And in presence of the enzyme that is called as succinyl co-synthetase, and this co-synthetase, what happens, it removes the coenzyme A, and with the removal of its coenzyme, it simply converts to succinic acid, which is a 4 carbon compound. So 1 carbon, 2 carbon, 3 carbon, 4 carbon. So this 4 carbon compound undergoes dehydrogenase reaction in presence of the enzyme succinate dehydrogenase by converting FAD to a compound that is called as FADH2. Got it? And this FADH2 conversion gives rise to a compound that is a fumarate. And this fumarate is otherwise, it is also a 4 carbon compound. So this succinate dehydrogenase is the only enzyme found to be present in the, that is, inner mitochondrial membrane. It is in inner mitochondrial. It is found to be present in the inner mitochondrial membrane. So this fumarate, it is also 4 carbon compound, carbon 1, carbon 2, carbon 3. In presence of the enzyme fumarase, it accepts the water molecules. In presence of fumarase and water molecules, a 4 carbon compound acid is formed that is called as malic acid. And this malic acid undergoes dehydrogenase reaction, that is in presence of an enzyme that is called as malate dehydrogenase. In dehydrogenation, what happens, an NADH2 is being produced with a 4 carbon compound, that is oxaloacetic acid is produced, which is the parent compound which is initially used in the cycle. So by this mechanism, it has been found that in total Krebs cycle, that is the cycle has been completed, and it was given by the scientist that is called as Kreb, that is called as the Hans Krebs, so it is called as the Krebs cycle. And what happened, this part, from this part to this part, it is a reversible reaction of 6 carbon, and from this part to this part, it is a reversible reaction of 4 carbon. So there is a 6 carbon reversible reaction taking place and a 4 carbon reversible reaction taking place. And here we find this is a 5 carbon, it is only 5 carbon compound present here. Is a complex reaction, a gateway, and another gateway we produce, that is the link that is produced, that is called as the this link, that is the pyruvate conversion of pyruvate to the 2 carbon compound, that is acetyl coenzyme A. So all we have to know is that in this reaction, this total reaction, what happens, starting from oxaloacetic acid, the cycle continues, and here is one complex reaction, and here is also one complex reaction. is the two complex reaction taking place, and I'd like this part is the 4 carbon reaction, this part is the 6 carbon reaction, and both of them is the are regarded as the total, that is called as the 6 carbon compound reaction, this part, and this part is the 4 carbon compound. In total, that is, this is the completion of the Krebs cycles. So what happens next? Move on. So during the Krebs cycle, what happens? What are the total, that is the Krebs cycle, and what happens? As we know, that is, as we know, in the mitochondria, and this is the cytoplasm, and in the, as we know, this here glycolysis takes place and here Krebs cycle takes place. So as a result, total aerobic respiration, so total, that is aerobic respiration takes place, that is in not only in the, that is called as in the cytoplasm, plus in the, that is mitochondrial matrix. So Krebs cycle plus glycolysis, so the total in Krebs glycolysis, total 8 ATP is being produced, and in Krebs cycle, total the link in the link reaction, that is 6 ATP is produced, and in the Krebs cycle there are 24 ATPs are produced, so total 38 ATPs are produced in them. So how subtle, with the help of subtle reaction, there will be 36 ATP produced. I will explain it later, but for time being, it has been found only 38 ATP is being produced.